So, Albie and I have been working together on mouse models of Alexander disease for several years now. And today I'm going to talk about a new rat model, which we're pretty excited about because it's coming together at the same time with some very promising therapeutic strategies that Ann Smith is going to talk about this afternoon. So we're excited about it, and I'd like to just give you a brief introduction. So I'll start with a little bit of background, and you've heard some of this already today, but so as most of you know that as you should know by now, Alexander disease is caused by mutations in glial fibrillary acidic protein. And we can't quite get away from this nice diagram I think Ali made of the location of the mutations in the protein. But one of the things I wanted to emphasize in this slide is that GFAP is an intermediate filament. It's a structural protein in astrocytes in the brain and spinal cord, and if you just put GFAP in solution, by itself it will form these nice filaments. It kind of self-polymerizes. And here you can see a GFAP stain in brain, and again you can see GFAP forms these nice, it fills the processes, the cells are very, they're kind of star-shaped, and you can see GFAP staining going out into the processes. Astrocytes are called astrocytes because they're star-shaped. And when the protein is mutated, and again these are missense mutations as Jim described, it disrupts the ability for GFAP to form these nice filaments. Not completely, they can form filaments, but they're more like, but they also form, I think we've heard them called blobs. I like to call them clumps. But you can see here again, you've seen the pictures of electron microscopy where you have GFAP aggregates, and these include other proteins that are intermediate, including other intermediate filaments and stress proteins and other cytoskeletal proteins. And then you can see, if you look closely, that they're kind of embedded in a meshwork of intermediate filaments that seem to be snagged in them. And so, again, I'm sure many of you are all too familiar with the symptoms, but Alexander disease can be divided into two classifications based on the location of the lesions in the central nervous system. And I think it's important to go over some of the symptoms because when you're making a model, you want to know, what you're looking for in terms of features in the animal. And so type 1, as we all know, has more forebrain predominance, and the classical symptoms that we associate with early onset leukodystrophy, and this would include seizures, macrocephaly or enlarged heads, cognitive and motor delays, and just a general failure to thrive. And then the type 2 disease has more hindbrain predominance, and it can occur at any point during your life, and the symptoms tend to match where the pathology is, so they're more bulbar or brainstem symptoms, which include difficulties walking, problems with speech, problems with swallowing, eye movement disorders, palatal tremors, things that involve your cranial nerves. So when we're thinking about GFAP mutations and the things that are happening on the molecular level and how these end up manifesting in what we see as Alexander disease, we've made several different model systems to try to understand how you get from one point to the next. And you've already heard a little bit about some of the model systems from Mel. And originally, some of the models were basic cell culture systems where we tried to overexpress mutant GFAP in cell lines. And Sushan Zheng is going to talk about cell lines that are actually derived from patients later on this afternoon as another model. Mel talked about the fruit flies, which we've done a lot of work to, she's done a lot of work to understand mechanisms, the underlying molecular mechanisms of the disease. And of course, our work has revolved mostly around mouse models of the past years. And all these models together have contributed a great deal to what we know about the underlying pathology in Alexander disease. And so I guess the question here is, why do we want to make a rat? And I think we're at a point where the mouse models just really aren't sufficient. They replicate many of the things that we know about the cellular and molecular pathology, but on a behavioral level, if you're looking at symptoms, they have very subtle cognitive deficits and no motor deficits, and they're really pretty normal just to look at them. So for the rat, it's been shown with some other disease models that if you make the same mutation in a mouse and a rat, the rat symptoms seem to more closely resemble what you see in humans. And so some of the other advantages would be that historically, many of the behavioral assays that scientists perform were developed in rats. And so rats are more amenable to some of the tests that we have to analyze how they function, both in terms of motor function and cognitive tests. And rats, if you work with them a little bit, they're smarter than mice. I wouldn't say that there's not really a comparison with humans, but certainly they're better at performing the cognitive tests. And of course, they're bigger. So Mel's point, there's definitely advantage to a small fruit fly in terms of economics, but if you're studying things that you'd like to assess in patients, you can't really look at brain very easily, so you're probably going to be wanting to look at things more like body fluids, such as plasma or cerebrospinal fluid, CSF. And in the rat, those are much easier to collect, and you can do it over time in a rat compared to a mouse, where if you're trying to collect CSF in a mouse, you get about 10 microliters, which is not much to analyze. And so the last point, I think for me, has been the most important. What can you look at in terms of a clinical improvement? What measures can you look at to say, hey, this rat actually looks better? So what measures can we look at to look at behavior and function? And so those are things we're hoping to have improvements for with the rat model. And so again, the diagram with our nice series of mutations. And I think it's been mentioned that this probably isn't up to date, but Mel pointed out that there's a couple of hotspots for mutations, one at arginine 79 and one at arginine 239, and we've made mouse models at both of these locations. And the rat model, we wanted to target the arginine 239 to histidine. This is a mutation that's in patients is predictive of a severe clinical course. And so this is what we chose to model in the rat. And we used CRISPR-Cas9 targeted mutagenesis. And this is a bacterial system that you might have heard in the news recently. It's actually part of the bacteria's immune system and how they ward off viruses, but we've co-opted it to if you've heard of it in the news, it's oftentimes referred to as maybe a potential therapy to correct mutations, but in our case, we've used it to generate one. And so we can target specific nucleotides, and this is showing a DNA sequence, and the nucleotide change that we've made results in an arginine at the position 237 to histidine. So I'll refer to this mutation a few times throughout the talk. But it's the same arginine as this 239, just in the rat sequence. It's offset by a few amino acids that are missing from the beginning of the protein, but it's the same arginine. And so the first thing you notice when you look at the rats is they're considerably smaller than their normal wild-type litter mates. These two animals are 8 weeks of age, and normally this is an adult rat, and they can be used as breeders, and our little guy here is about 1/4 of the size. And if you look at them developmentally and using body weight as a basic measure of their condition, up to about 3 weeks of age, the mutant animals are indistinguishable from their wild normal litter mates, wild-type normal litter mates, non-mutants. And after that, they don't progress as well. They don't gain weight like their litter mates do, and actually they start to lose weight. And by 8 weeks of age, they're in pretty bad shape. They become ataxic, they're wobbly, their gait is unusual. We've had some animals that actually became paralyzed. These animals are having absence seizures at this point, multiple absence seizures, an average of 40 per hour. Is that right? This is work in Micah Niedergard's lab that she's shown this with EEG analysis. And so they're not doing very well at this point. But they do start to go through what you might call a recovery period, where they start to gain weight and they get big enough where they can, they're fertile. Females can carry litters, but they never quite get to the point of, a normal rat. They still are ataxic, they still have problems, and they still have a lot of pathology. So if you look at the brains at this time point, at 8 weeks of age, this is looking at GFAP in the brain. And this is a normal animal, and you can see these nice wispy star-shaped astrocytes again. You can see the fine processes in these cells. And in the mutant animals, you just see this dark, dense staining where you can pretty much see the aggregates here that are surrounding the dense in the middle of the cell body, and you almost lose the ability to see the processes. If you look closely, they're there, but it's hard to see them. In this higher magnification image that's in fluorescence staining, you can see the shape of the cells, and you can kind of see the center. They're very globular, and this is all GFAP. And you can see the processes are thickened and kind of stubby looking, especially if you compare it to some, it's different stains, but you can get the idea. These look nothing like the nice star-shaped astrocytes I showed earlier. So Jim's point about the whole morphology being really messed up is true in the rats as well. The shape of the cell is way off. You can quantify GFAP, and here I'm showing two different graphs, one quantifying protein, and the other is quantifying RNA. So it's important to look at both, and I'll get back to that. But it might not surprise you to learn that GFAP is accumulating. We've shown you these images with EM where you see these clumps of protein, and so those aggregates accumulate. GFAP itself in filamentous form or in other forms is increasing. And here in red, these are different, these bars represent different brain regions. And so across the brain, regardless of what region you look in, the mutant animals represented with the red bars have much higher levels of GFAP protein than the wild types, which are in black. Transcript is also elevated. So this is RNA, which is encoded by the DNA, and it gets translated into protein. This is elevated too in the same brain regions. And this is important. Let me just backtrack a little bit. Astrocytes, as Jim mentioned, have some features of an immune cell. They're not a classical immune cell like the microglia, but they do have some features. And when you have an injury to the brain, they become reactive. And GFAP is one of the key markers, or one of the key proteins that gets elevated when astrocytes undergo this response. And this happens in traumatic brain injury, inflammation, if you have infections. It's a generalized response. And so in the case of Alexander disease, you have this toxic mutant protein that's being produced that's causing a stress response to the astrocyte. So what does the astrocyte do? It upregulates GFAP. So you get more production of GFAP, and we see that at this RNA level. And so it's a nasty positive feedback loop that the astrocytes keep synthesizing this mutant protein. And I wanted to mention that because if you're going to stop Alexander disease or do something to ameliorate the problems, this might be a good place to start. So if you look at GFAP accumulation over time, I showed you the body weights earlier. And so this is the same graph, and just to remind that around 3 weeks of age, the animals look pretty normal. They do have the Rosenthal fibers, the aggregates. You can see that histology in the brain if you look. And this is looking at protein directly on a Western blot, and we've got wild-type animals here represented on the left, mutants on the right. This is 3 weeks of age, and we're beginning to see an increase in GFAP protein here. And then at 8 weeks of age, when the animals are very sick, we see massive accumulation of GFAP protein. So it kind of matches what we're seeing on the molecular level compared to what we're seeing in the symptoms in the rats. And these are other proteins that are found in Rosenthal fibers. And again, we're looking at the animals at 3 weeks of age and 8 weeks of age. And we begin to see an increase in the mutant animals at 3 weeks of age. And then at 8 weeks, all of these proteins, which include vimentin. This is another intermediate filament. I mentioned those are part of Rosenthal fibers. Small heat shock proteins, which are stress response proteins. These are also components of Rosenthal fibers, and other stress proteins are elevated. And this is important to show because it validates the model. These are things we predicted would be elevated based on studies of human tissues and our mouse models, and we see that they're also elevated in the rat. And of course, they have the clumps of protein. This is electron microscopy again, and you see the same pattern where you see these clumps of protein. This is an astrocyte, so it's filling up the cytoplasm here. In a closer look, you can just see that they're embedded in this meshwork of intermediate filaments, and this one, it almost looks like it's radiating out of it. So all of this goes towards the model seems to be working as we would predict it would. We're seeing that the astrocytes are becoming stressed and having a gliotic, what's called a gliotic response or gliosis. We see elevation of GFAP at the transcript level and at the protein level, which is typical of reactive astrocytes. We see elevation of stress response proteins, some of which are included in Rosenthal fibers. And we've shown that they have Rosenthal fibers with electron microscopy. But the big question, which I think I started off with, is how do we, let me see if I can get the question out here. Do they show symptoms of the disease? Because I think that was what we were missing in the mouse models and the whole purpose of making the rat, generating the rat. And so this is a test for motor deficits. It's pretty aptly named. It's a horizontal ladder test. And you can see here on the left, this is a normal wild type rat, hopefully. Give me a second. It should go. It went earlier.
Can you tell it?
I'm afraid let's see. Sometimes if you keep clicking it just goes fast forward through it. That's what I was afraid of. Let's try this one more time. Oh, shoot. Sorry, I really, well, I don't think it's going to go, so.
But you can see Tracy in the background.
Yeah, you can. That's nice. Shoot. Well, that's too bad. So if you put the wild type rat on here, he makes it across pretty rapidly. And the test is valuable in that you can score the number of times that their paws slip in between the rungs. You can score how quickly they get across it. And when you put the wild type rats get across there pretty easily, and they get a nice treat at the end, which you don't get to see. But anyway, the mutant rats, you can see this little guy here. You can see how he's shaped. They really struggle with this test. Their paws slip in between the rungs. Many of them, you give them a minute to get across the ladder, and they basically, some of them you have to take off, you stop the test early. Some of them make it across, but the test is really too difficult for them. And so a simpler, still not gonna go. Okay. A simpler test is just a basic test of grip strength. And you can see here that this rat is being tested for hind limb grip strength. So this is just a force meter with a little bar attached, and you allow the rat to grab the bar and you move it away from the meter, and you can measure how much force it can apply to the bar. And you can measure hind limb grip strength is what's shown here, or forelimb grip strength. And you can see that the mutant animals shown in red here are a lot weaker than their wild type counterparts. And another test you can do to look at coordination is the Rotor Rod Test, which again is somewhat self-explanatory. It's a rotating rod, and it starts off slow at four revolutions per minute, and then it ramps up to about 40 revolutions per minute over a 5-minute period. And you score them for when they fall off. And it's automated, so the machine will give a readout for how many seconds they stayed on here, and you can see that wild type animals, over the 4 trials that you put them on the rotor rod, will gradually improve, and some of them actually will stay on the rod the whole time. But the mutant animals basically can't stay on the rod, so in about 30 seconds they fall off. And the reason that, to me, this is exciting, because this gives a really big window here for us, if we're going to test therapeutic strategies in these animals. This gives you a lot of room to look for improvement. And so this is what we were missing in the mouse, and I think we have it in the rat. And so I'm going to go ahead and wrap things up. So in summary for the rat, this work is in progress, but we've basically shown that we mimic the molecular and cellular pathology of the disease, at least what we've shown so far. And more importantly, we've demonstrated that there are clinically relevant symptoms, including a failure to thrive, and motor and cognitive impairments. I didn't talk about some of the results we have from cognitive behavioral tests, because they're very preliminary, but we believe they also have impairments with cognitive function. And so future directions are going to be using some of the therapeutics that will be talked about this afternoon, and some of the other studies that, for example, with Mel talking about with the fly studies, to see if we can prevent or reverse some of these features, not only the molecular and cellular features, but some of the clinically relevant ones. And with that, thank you for listening, and obviously thank you to Albie Messing, who I've enjoyed working with for the past many years and on these models together. And as Mel mentioned, I think has been a huge driving force behind everything that you're going to see today. And Denise Springman, who takes wonderful care of our mice and rats, is retiring this year, so she will be missed. Ming DePern, who is here from Taiwan, helped with much of the protein work that you've seen today. And then, of course, thank you to all of you and the families of patient groups. This work would not have been done without that, and that includes the Juan McBun, Elisa's Corner, Klein, and the United Leukodystrophy Foundation. Thank you.
Okay. Questions?
Have you been able to use an ASO in your rat model yet?
We're working towards that. So they do match the sequence, and it's fortunate that because the ASOs are sequence specific towards the mouse, that we do have some overlap with the rat sequence. So we do think they're going to work in the rat. So that's the next step. So that's why we're really excited that these things are coming together. Hi.
When they have like the seizures and stuff, do you guys treat them with any sort of medication to kind of try to slow seizures down so you can see the progression of the disease without the seizures?
We have not done that. I think there have been some tests. It's very preliminary. This is work that's been done in Micah Niedergaard's lab, who will be talking this afternoon, but I don't think she's talking about seizures. I don't know if you
can treat the seizures.
So yeah, she just mentioned that they can treat the seizures. So there are ways of ameliorating those. So those are things we could look into in the future. Yeah.
Yeah, because I feel like kids like seizures, every time they have one, they're losing something, you know, so like mice or rats.
Hi.
I just have a question about if there's any barriers that you guys are experiencing that there's something that we as families, friends could do to help move the whole process along, or is it just a matter of getting the machine rolling?
So I forgot we're supposed to be repeating the questions, but are there any barriers to moving things along? And I think all of us are working really hard to get this, to keep this progress going. And Ionis Pharmaceuticals has been really integral, and it's a really nice collaboration because there's been a push back and forth. And so I think between all of us working on this, I don't know, maybe Albie could answer if there's a other
Yeah,
elements.
so, I think, maybe I should go over the microphone. So so I'm glad you asked, although it's a really complicated question. I think in some ways, in many ways, a lot of what we have done has been guided by the information that has been shared with us by patients and families about the nature of their disease, their experience with progression of symptoms, and the participation in the natural history study that's going on at CHOP now is just a much more systematic, basically, and thorough way of gathering that information and putting it together in a way that's going to be really useful in planning clinical trials. I'd also say that we've been very fortunate over the years in being funded by the NIH for what Melfini mentioned as the program project, which is really focused on a lot of the basic mechanisms of the disease, the kinds of mutations that cause disease, and trying to work out all the downstream pathways that are involved. But frankly, the effort aimed at developing treatments has largely been funded by donations to research. And that's because it's challenging to get any grants from the NIH, but it's even more challenging to get ones that are that specifically aimed at developing treatments. So that's something we always appreciate, yeah.
So after, obviously you guys have done the flies, the mice, and the rats, after the mice, or the rats, do you go directly to humans, or do you try like a pig or something?
I'll answer that.
We thought about pigs, but Albie's no on the pigs.
So, I just spent 2 days at Food and Drug Administration as part of a conference where Alexander disease was fortunately included as one of 5 case studies for how to go about thinking about, taking an experimental therapeutic and moving it forward into clinical trials and use. And one of the questions that came up was the value of model systems, and how many model systems do you need, and how perfect do the model systems have to be. And the real interest in Alexander disease for that group was just the fact that it's so rare. It's not like hypertension, it's not like high cholesterol. It's really rare. There aren't that many patients where you can collect data or test therapies on, and so so I think one of the messages was that it's really helpful to have several model systems like what we're doing that complement each other and can inform what you're going to do. But even though they're not perfect, flies don't have myelin, the rats don't have, even mice and rats are close but not completely reflecting all of the human pathology and symptomatology. Even though they're not perfect, it's in this case they seem to think the case for a treatment is strong enough that you could go right to human. Now, in theory, you could make pigs. You could even make primate models. Those become exponentially more expensive to make, to maintain, and to study. And so then it's just a calculation. Do you really want to wait and do that, or maybe it's time to just go right into human and try it there? So I think at least that's our thinking about it. Also, I'm afraid of rats. I'm not sure what I would do with a pig. So, yeah.
I know it's probably premature, but right to try accelerate things, since most of our kids have a short
Yeah.
span.
So the question was about the right to try. Is that a law, or maybe I'm going to let Amy Waldman try to answer that.
Can I say it here, or do you want to know?
You can stand up.
Okay. So
the right to try, I think, is something as physicians and scientists we all completely understand and respect the fact that in our country we have access to things that other countries and stuff do not. I think it's quite complex, though, from a scientific standpoint, because it breaks the laws of how to prove whether a drug is safe and whether it works. So one of the quotes that I love from, it came out of the laws that say that drugs need to be studied in pediatrics. One of the quotes was, which is better, a well-designed clinical trial where there's certain measurements at certain times that you're investigating the safety and efficacy in a pre-planned, well-designed way, versus an N of 1, I mean a trial in just a single patient, and giving something that we don't have proven safety and we don't have proven efficacy. And I think we all really do understand, as a parent, how important that right to try is, so that you know that you as a parent have done everything you possibly could to help your child or your family member. I think it does make the science a little more challenging on our end to demonstrate that Hippocratic oath that we've all taken to do no harm and to make sure that it is in fact safe and appropriate, and we're not going to do any harm to your child. So it's a little bit as a field. We're still trying to kind of grapple with this right to try, and whether or not how we're going to sort of incorporate that into our daily taking care of patients. I don't know if that answers your question, but
That answers the question, but along the same lines, can you take a control, someone that does not have any problems, that volunteers to try a drug like that to see if there's any side effects before you try it on our kids?
Well, that's a great question. I'm going to actually address that later in my talk a little bit. I mean, just to give you the very quick highlight, there are different types of trials, and there are trials in healthy volunteers, which is not something we generally do for a disease like this or a therapy like this. And I do also have to highlight the work that Ionis has done with another disease called spinal muscular atrophy, and using an ASO and generating safety data in something that has already now been FDA approved for a very terrible disease. So we're able to get some of that safety data, not from the same ASO. It's a different ASO, but just the technique and the stuff from this other disease model.
I thought with the right to try law that a drug still has to at least make it through phase 1, I believe.
Stage 1, yes.
Yes. Okay.
And that's where it gets a little bit complicated, because sometimes in rare diseases we don't do a standard phase 1 trial. But yes, there are some rules in terms of the right to trial.
Question actually for Tracy. Do you,
See if I can answer it.
do you always inject the rats at birth, or have you ever done more than older rats to kind of imitate like the, not the infantile form, more of a juvenile or
When you
onset?
So when you say inject them with...
Well, have them imitate the disease.
Oh, so-
Do you ever have them imitate a little bit older instead of not at birth?
The mutations were generated originally in a fertilized egg, and then the animals, you generate a first generation of animals from that, and then, so those animals automatically have the mutation, and then it's inherited after that.
So you can't do it while the animals are...
Not with what we're using here. There are ways to do that, and we haven't tried it. You can use viruses to modify genes in the brain. That's not the technique we're using, and with this model we probably wouldn't go that route. There may be things we could do to inhibit GFAP expression using the ASOs, and then allow that to wear off and then see what happens. But those would be experiments in the future, so I don't.
Is that the same thing as the fly models do?
Similar, different techniques.
That's in the womb and not outside?
Yeah.
Okay, so we'll take one more question.
Yeah. I'm trying to remember the paper that came out in December on the mouse model and the ASOs. I'm trying to remember, it's been a while since I looked at it. Were the ASOs given at different points in the mouse's life to see what would happen? And if so or if not, will you consider that in the rat model rather than just giving all the drug at the beginning, maybe midlife?
Yeah, so Ann Smith is going to talk about that study in a little more detail, but why don't you just answer that particular question?
So with the mouse models, we generally injected them at one stage when pathology had already developed. And again, the mice don't show severe clinical symptoms, so that wasn't an aspect we could look at. With the rats, if we're going to treat the rats, then it would be nice to treat them at an early age, say that 3-week time point when they're really not showing much of an effect, to see if we can prevent the symptoms. And at that later time point, which might be more analogous to a patient who's been diagnosed, because, by the time you get a diagnosis, can we inject a very sick rat and see if they're going to show recovery? So that's really a big goal, and looking at both of those would be important, I think.
And I think that's maybe later on I'll have a question. I think thank you, Dr. Waldman, for answering my email months ago. I don't know the FDA process, but as a parent, I know that the clinical metric is really significant, and generally that needs to show improvement. But I guess maybe we can talk about this later. But as a parent, in a metric, stabilization. If somebody's declining and now they're stable, to me that's a change.
Good thing, yeah.
And stabilization as a parent is just as important as actual improvement in the metric. So
I think that's why the rat model, that's why we're excited to have it, is because I think a lot of the tests that would be considered preclinical that we can do in rodents will be very informative for future studies with the new compounds that they're going to generate for humans. So I think hopefully we can make some comparisons there and some substantial contributions to what the work goes on in the future. Thanks.
Okay, so let's move on.